Gene cloning. Complete explanation. What is gene cloning? Gene cloning is a molecular biology technique used to produce identical copies of a specific gene or DNA sequence. It involves inserting a gene of interest, GOI, into a cloning vector and introducing it into a host organism, typically a bacterium, where it can be replicated and sometimes expressed. Objectives of gene cloning: to amplify DNA fragments to study gene structure and function to produce proteins, e.g., insulin, growth hormones, to create genetically modified organisms, GMOs, for gene therapy and vaccine development. Steps in gene cloning process. 1. Isolation of gene of interest. The gene is isolated from genomic or complementary DNA using restriction enzymes, endonucleases. These enzymes cut DNA at specific recognition sites to yield sticky or blunt ends. 2. Selection of a suitable cloning vector. A vector is a DNA molecule used to carry foreign genetic material into a host. Good vectors must replicate independently, contain a multiple cloning site, MCS, have selectable markers, e.g., antibiotic resistance genes, common vectors, plasmids, bacteriophages, lambda-phage, cosmids, BACs, bacterial artificial chromosomes, YACs, yeast artificial chromosomes. 3. Ligation of DNA into vector. The gene and vector are cut with the same restriction enzyme to ensure compatibility. DNA ligase enzyme is used to seal the gene into the plasmid, forming recombinant DNA. 4. Introduction into host organism. The recombinant plasmid is introduced into a host cell, commonly E. coli, using transformation techniques, heat shock electroporation, microinjection, gene gun, in plants. 5. Selection of transformed cells. Not all host cells will take up the recombinant plasmid, so selection is necessary. Antibiotic resistance genes, e.g., AMP-R, reporter genes, e.g., LACZ for blue-white screening, cells with successful recombination will survive or change color on selective media. 6. Screening and confirmation. Colonies are screened for the presence of the desired gene using PCR restriction, digestion sequencing, colony hybridization. 7. Cloning and amplification. The transformed host cells are cultured in large numbers. As they divide, they replicate the recombinant DNA, thus amplifying the gene. Tools of gene cloning. Tool function. Restriction enzymes cut DNA at specific sequences. DNA ligase joins DNA fragments. Plasmids, vectors carry foreign DNA into host cells. Host organisms express or replicate recombinant DNA. Selectable markers identify successful recombinance. Applications of gene cloning in research: gene structure and function studies, functional genomics in medicine, production of recombinant proteins, e.g., insulin, interferon, gene therapy for genetic disorders. DNA vaccine development, e.g., for COVID-19, in agriculture, creation of transgenic crops, e.g., BT cotton, golden rice, pest and disease resistance, enhanced nutritional content in industry, production of enzymes for detergents, brewing, etc. Biofuel production, ethical and safety concerns, GMO regulation by FDA, USDA, and EPA, biosafety levels for handling recombinant organisms, bioethical debates on human gene cloning, designer babies, key concepts, recap, concept description, GOI, gene of interest to be cloned, vector DNA vehicle to carry and replicate GOI. Host organism that replicates the recombinant DNA, clone genetically identical copies of DNA, gene, cell flowchart of gene cloning process, C#